Okay, we're going to go on to the third part of our recombinant DNA technology lecture, and in this lecture we are going to focus on cloning. So we already talked about the plasmid, and the plasmid is a key part of recombinant DNA technology. So you already know plasmid is a double-stranded circular piece of DNA that commonly has some sort of selectable marker, which is usually an antibiotic resistance gene. There is an origin of replication at some point on the plasmid, and you have your multiple cloning sites. And those multiple cloning sites are those sites where you can insert foreign DNA into the plasmid. Okay, so here we have a plasmid. We have the antibiotic resistance gene that's in this plasmid. We have our foreign DNA, and usually it's a gene that we're interested in studying, a gene that has some sort of function that we want to look more closely at. So you're going to cut, using your restriction endonucleases, both strands of the foreign DNA of interest. You're also going to use the same restriction enzymes to cut your plasmid. You are then going to use the enzyme ligase to glue in your foreign DNA of interest into the plasmid, and both should contain the same corresponding ends. In this case, it's sticky ends. You end up generating your recombinant plasmid. You have your plasmid DNA containing your antibiotic resistance gene and this foreign DNA of interest, this gene that you want to study. Okay, great. You've got this recombinant plasmid. What do you do with it? You can't just have it sitting there in a tube and expect it to do something. So what you want to do is generate multiple copies of this plasmid. So we already said that plasmids are autonomously replicating. They can replicate on their own outside of a chromosome. So what you need to do is generate many, many copies of these plasmids by putting them into a host cell. So there are various types of host cells that you can use in cloning. One of the more common being bacteria, and specifically the Escherichia coli bacterial cells. So you can place your recombinant DNA, your plasmid, using heat or using chemicals. You can force the plasmid into your bacterial cell, and your plasmid is going to sit in here inside the cell next to the bacterial chromosome. And you all know that bacteria will very rapidly, E. coli every 20 minutes will divide through binary fission. So one cell becomes two, two becomes four, and very quickly you have many, many, many copies of your recombinant DNA, your recombinant plasmid. Then what you would want to do is purify your recombinant plasmids from your bacterial cells. They have these plasmid purification kits where you can purify plasmid from bacterial cells in about 10 or 15 minutes, and you end up with a tube containing purified recombinant DNA. So again, you have your bacterial cell. Your bacterial cell contains your plasmid. It contains your bacterial chromosome. You generate many, many, many copies of your plasmid through typical bacterial replication, through binary fission. And each one of these organisms is called a clone. And that is why this whole process is referred to as cloning, because you are making an identical copy of your recombinant plasmid using a host cell, and in this case it's bacteria. Now the nice thing about using bacteria is that bacteria grow very quickly and very inexpensively using an agar media. So what you would do is you take your plasmid, your recombinant plasmid that contains the foreign piece of DNA, and as you know, the recombinant plasmid usually also contains a antibiotic resistance gene. So when this plasmid is grown on agar media that contains the antibiotic that is in your plasmid, so let's say this antibiotic resistance gene right here is the ampicillin resistance gene. If you streak your bacteria out onto a typical nutrient agar that contains ampicillin, there's ampicillin in this agar, the only bacteria that are going to be allowed to grow on this agar are bacteria containing this plasmid, because any bacteria that doesn't have this plasmid with the ampicillin resistance gene wouldn't be able to grow on the agar media containing ampicillin. So we wouldn't want to grow the bacteria in agar without any antibiotic, because what would happen is if in our tube, our culture tube that we have our little bacterial cells containing our plasmid, if we had some bacterial cells that don't have the plasmid, sometimes some of the bacterial cells don't take up the plasmid, those bacteria that don't have the plasmid would be able to grow on this agar. And then you wouldn't know which colonies on the agar are the ones that contain the plasmid with the antibiotic resistance gene, with that gene that you want to study, and which colonies on the agar are just bacteria without that specific recombinant plasmid in them. So it's much easier, and that's why we call those antibiotic resistance genes selectable markers, because we are specifically selecting for the bacteria containing the plasmid by adding the antibiotic. So that's a key component of recombinant DNA technology and cloning. So in summary, you have your plasmid. Your plasmid contains that multiple cloning site. This usually is a string of restriction enzymes so that you can use multiple restriction enzymes to generate your recombinant plasmid. That plasmid also usually carries some sort of selectable marker, that an antibiotic resistance gene usually, and it will have an origin of replication. You are going to take your restriction endonuclease, whichever one you need to use to generate your recombinant plasmid. It might be BAMH1, it might be EcoR1, it might be HinD3, it might be SAL1, it might be 2. So in this cloning site, this one site might be, let's say, HinD3, and another might be SAL1. You might need to use both, cut with HinD3 and SAL1. You are then going to remove, by cutting it, this piece of DNA out. You are then going to cut your DNA of interest with your SAL1 and your HinD3, so you have the appropriate ends on here. You're then going to use ligase to glue in your foreign piece, your gene, into your plasmid. They have corresponding ends on them. You are then going to transform your recombinant DNA into your host cell. Your host cell might be bacteria like E. coli. Your host cell could be a virus, a bacteriophage, a virus that infects bacteria. Once you transform your foreign recombinant plasmid into your host cell, in this case a bacteria, you are then going to take your bacteria, grow it on agar media containing your selectable marker, usually an antibiotic, whatever's in the plasmid, so that you know each colony has that plasmid in it. You are then going to isolate a colony from that agar containing antibiotic. You can then grow it up in liquid media that also contains the antibiotic, and you can purify your recombinant plasmids from that liquid media, and then you have many, many, many copies of identical plasmids or clones. So that's how cloning works. That's really it. It's actually a pretty simple process. Cloning, it can be very complicated, and you have to design everything very carefully. So you have to design your plasmid. There are many, many, many different types of plasmids and phages out there. So you have to choose the right one for what you're trying to do. Within the plasmid, you have to then choose the right restriction enzymes. You have to choose the ones that are going to work and are going to put your gene in the right orientation. Once you figure out the right enzymes, you need to then amplify your gene of interest. You want to get a lot of it. You need to also cut your gene of interest with your restriction enzymes. Your next step here would be then to ligate your DNA into your cut plasmid. Then you are going to put it into a host cell, so into your bacteria, grow it on agar containing your selectable marker. Then you're going to take your colony, grow it up into media, and purify your plasmids using a plasmid purification kit. And there's many companies that make plasmid purification kits. You now have exact clones. You can then go transform these plasmids into mammalian cells or any type of system you need. You could put them into an animal model, whatever you need to study that gene to characterize its function or utilize that gene to generate a product that you might want to purify, such as a vaccine or a specific enzyme used to treat a disease. So recombinant DNA technology and cloning is a very exciting and very readily usable technology in the molecular laboratory.